In this video, we're going to discuss antibiotic resistance. So, antibiotic resistance is a huge issue in microbiology. We're constantly seeing these newly evolved strains of bacteria that are resistant to all of the drugs that we have available. So, for example, a couple years ago, a man was diagnosed with gonorrhea, and they actually found that the strain of Neisseria he was infected with was resistant to every single antibiotic that we have available. So how does antibiotic resistance happen? Okay, so let's say this patient, they have this pathogen. All right, so here's the pathogen. And now there's always going to be in this community a couple of bacterial cells that are kind of mildly resistant. Okay, they're mildly resistant to the antibiotic. And meaning that the antibiotic will eventually kill them, but it'll take longer than the rest. Okay, so now this patient is prescribed some sort of antibiotic and they begin taking it. So day one, it's going to kill off some of the pathogen. Day two, we're going to kill off some more. Now by day 3, this patient is feeling so much better. They feel fine. They don't feel sick anymore. And guess what they do? They stop taking their antibiotic. They don't take it as prescribed. And now guess what can happen? We still have some of those mildly resistant bacteria that have yet to be killed by the drug. So those guys can start now growing in the body. And from there, we're going to get some even stronger, severely resistant bacteria in there. All right, so these guys are full-blown resistant to the drug. Now the patient may have a reoccurring infection. They may feel sick once again, and the doctor may give them the same antibiotic. And of course, this time that antibiotic is no longer going to be effective because all of these cells have now gained resistance to the drug. So this is a common issue we see, especially with patients that don't adhere to the advice of their doctor. Now, in terms of the actual genetics, right, how do the bacterial cells gain those antibiotic resistant genes? And there are 3 ways bacteria can pick up antibiotic resistance. Okay, so the first way is called transformation. Transformation is pictured here. Okay, this bacterial cell has died. When it died, all of its DNA got released into the environment. And it happened to have a gene here that's coding for some antibiotic resistance. Okay, so it happened to have an antibiotic resistant gene that's now naked in the environment. What can happen is this gene can be taken up by another bacterial cell. All right, so this new bacteria has taken up this naked DNA from the environment, and it has now inserted that antibiotic resistance gene into its genome. Okay, so during transformation, it's the uptake of naked DNA from one cell to another. The second way antibiotic resistance can spread is through conjugation. We've seen this before, okay? Conjugation. Conjugation is where one cell, okay, this cell here, this cell has a plasmid, and on this plasmid is a gene for antibiotic resistance. So it has an antibiotic resistance gene on it. Now during conjugation, one cell can transfer the plasmid that contains the antibiotic resistance to a neighbor. Okay? This process, conjugation, is very common, especially in hospital environments. Because in the hospital environments, a lot of sick people are coming in that have these bacteria, that have these plasmids, and then they get in contact with other people or at the bathroom or in the food, okay? And then very easily these plasmids can be transferred from cell to cell. So conjugation, transfer of plasmid DNA. And then the third way antibiotic resistance gene can spread is called transduction. So transduction is when a virus, okay, so there are some viruses that can infect bacteria. And sometimes these viruses can pick up an antibiotic resistant gene from one bacteria, and then the virus can go on to infect another bacteria, and in the process it can transfer the antibiotic resistance from one cell to another. So altogether, transformation, the uptake of a naked DNA that codes for antibiotic resistance; conjugation, the transfer of a plasmid; or transduction, the transfer of a virus that's carrying an antibiotic resistant gene from one cell to another. Now that the bacteria, this new bacteria, has a gene coding for antibiotic resistance, what do these genes do? Okay, how do these genes allow the bacteria to survive in the presence of an antibiotic? Okay, and there's 5 ways that the gene can allow the bacteria to survive. The first way, the gene can code for an enzyme. So the gene codes for an enzyme, and the enzyme works to destroy the antibiotic. So I'll give you an example. We've talked about this bacteria before called MRSA, all right, methicillin-resistant Staph aureus. MRSA is resistant to methicillin, but they're also resistant to penicillin. All right, so if someone takes penicillin to treat this staph infection, the bacteria actually release these enzymes called penicillinase. Okay, penicillinase. It's an enzyme that will destroy penicillin. So when they release these enzymes, they target the penicillin and they chop it all up so that the penicillin can never actually get into the cell. It blocks it from ever coming in because it will destroy it before it can come in. So that's the first way the bacteria can destroy the antibiotic, using an enzyme. The second way the bacteria could resist the effects of the drug is they can block entry of the antibiotic. All right, so basically how this works, all bacteria have normal receptors here. Okay, they have a normal receptor that normally would allow the antibiotic to come in. All right, so pictured here, a normal receptor that allows the drug to come in, and once the drug gets into the cell, it can quickly destroy and kill the cell. But some bacteria, when they get these antibiotic resistance genes, they change the receptor. Okay, so they change the shape of the receptor, so now the drug can no longer get in. It's blocked from coming in, and if the drug can't get in the cell, then the cell can survive. They're resistant to the antibiotic. The third way the bacteria can resist the effects of the drug is they can basically pump out the drug quickly. So they pump the drug out. These pumps, they have a special name. They're called efflux pumps. All right, so again, how this works is the receptors are still there. The drug can still come into the cell, but as soon as the drug gets in, the bacteria quickly spit it out really fast before the drug has time to work, using those efflux pumps. The fourth way the bacteria can resist the effect of the drug is they can alter the drug target. So, for example, if the drug normally attacks the ribosome, all right, and destroys the ribosome so that the bacteria can't make proteins anymore. But sometimes if the bacteria has now acquired genes, they can alter the binding site where the drug normally binds. Now the drug can't get in and bind, and it's basically useless. It doesn't do anything, and the bacteria can survive. The final way the bacteria can resist the effects of the drug is they can use alternative metabolic pathways. So use alternative pathway. So this works if the drug normally targets a pathway. So if the drug targets the pathway, if the drug normally targets the pathway and inhibits it from working, okay, to make a certain product, the bacteria just say, I'll just use this other pathway instead. So it's okay if the drug blocks this one pathway, I can use the other one. So those are the 5 ways bacteria actually resist the effects of drugs. Lastly, the preventing antibiotic resistance is the responsibility of both the healthcare worker, the physician, and the patient. All right? So we have to consider physicians need to be more responsible about prescribing antibiotics only for bacterial infections, only if they know exactly what the infection is being caused by. At the same time, it's the patient's responsibility to take the drug as prescribed completely. If for some reason they need to stop the drug, such as if they have an allergy or if the doctor changes the prescription, the drug needs to be properly disposed of. Disposing of antibiotics into water sources is really dangerous because then we're getting antibiotic resistance in our water supply. Okay, and also it's the responsibility of society. We do overuse antibiotics for animals and things like that. So it's the responsibility of many people to try to limit drug resistance.